Hello, Dr. Chris Magryta here with Salisbury Pediatrics, beginning another educational series on children's health. Specifically this time, we're going to talk about the growing brain. So what do we want to know about the growing brain? Well, to me, this all dials back to the simple understanding that what we really care about right now is, are we growing the brains correctly within the framework of what ancestrally we're supposed to do, i.e., are we providing all the nutrients? Are we providing all the pathways, the best chance to form in order to have the best neuro health outcome for our children? So this conversation is going to be a deeper look into what's happening in modern society as it relates to the growing brain. Are we hitting all the upstream targets that we need to achieve optimal outcome? And not just the basic understanding, but really what is optimizable right now in modern society. So ultimately, we care primarily about immune disease trends. Are we seeing big changes in immune-mediated disease and the ones that we worry about related to the brain? And we know now that immune-mediated disease in general across the board is rising exponentially for decades in children. And we don't see any signs of slowing down. So for me, the big problem right out of the gate is we have an issue with immune solvency. And the activity of the immune system is at the center of all the concerns that we have. What is the prototypical neurological concern of modernity? It is autism spectrum disorder. When I was born in 1970, it was around 1 in every 5,000 children. Some studies show 1 in 10,000, but I just suffice to say it was 1 in 5 to 10,000 range. So pretty significantly low compared to today. Now the incidence is lower even now than 1 in 38, which was the last number specifically noted at the CDC, but in some states we're seeing even down as low as 1 in 22. That's over 125-fold change in 50 years. So that's not an error of genetics. Genetics don't shift in a rapid speed like that. This is clearly a problem of an epigenetic nature, or something environmental is affecting how our DNA is being read or how we are turning into ourselves. What are the possible thought process here? What are the etiologies? In functional medicine, we talk about antecedents, the upstream reasons why something is happening. How do we go upstream to better understand this process? So we start to think about predictive modeling. And a fascinating study came out in 2023 at Duke University, where they looked at the predictive value of early autism detection models based on electronic health record data collected before 1 year of age. So at that point, they're saying, okay, we're looking at data before most people even realize a child has any issues related to neurodevelopmental changes or neurodiversity, like we call autism spectrum disorder. And they collected data on over 45,000 children, of which 924 or 1.5% met autism criteria. Model-based autism detection at age 30 days, mind you, and this is the key, 30 days achieved a 46% sensitivity, 23% positive predictive value, and a 90% specificity. So 90% chance that if they got it, it was right, about 1 in 2 chance that they had the opportunity to pick it up, and the positive predictive value being 23%. Then they increased that even more by detecting at 1 year. Their sensitivity increased from 46% to 60%, and their specificity went down a little bit to 82%. But ideally, you start to note here that there's a predictive ability to see upfront before we see the phenotypic changes as what's going on, because they started looking at other soft signs, hypotonia. They started looking at other related findings and did a machine learning algorithm to see if this was pre-predictable. And the answer was, it is. So for me, that brings the story all the way back to the beginning. If you're able to predict in a 1 in 2 chance that a child has autism spectrum disorder at 1 month of age, that is essentially telling us that the vast majority of the insults that are happening that are related to changing in the genetics or changing the outcome of the phenotype of the child is happening in utero primarily, and potentially in the first month of life or subsequently as well, but primarily in that early stage of the first 10 months of pregnancy. That to me is what this study shows, and it's fascinating. What do we know about nutrients, right? Is it only a deficiency state that matters, or maybe suboptimal energy levels as well? This is a complex question, but we do know one thing now after multiple studies show that maternal health matters a lot, as we shall see. One of the simple ways to know this is just look at epidemiologic data. If a mother is overweight by obesity criteria, during the pregnancy period, her child has a 2x risk of developing autism spectrum disorder. If a mother has diabetes as well as being obese, that problem then unfortunately will then fall into a 4x category increase. So therefore, we know nutrition has something to do with this, or at least nutritional-induced metabolic changes. And then we're looking at a study by Edlow et al., in this case, in JAMA, Network Open, where they started to notice that there was a sex-specific neurodevelopmental outcomes among offspring of mothers with SARS-CoV-2 infection during pregnancy. And we're hearing now is, from this data set, that maternal SARS-CoV-2 positivity was associated with statistically significant elevation in the risk of developing autism spectrum disorder at 1 year of age. And the odds ratio was 1.94, which is pretty high. So here we have a piece of information saying, okay, maternal infection drove disease. What's going on there? Well, something's going on immunologically. We'll talk about that. And the third big category that I want to touch on is maternal autoantibodies. How about if mom has antibodies in her bloodstream that are potentially biomarkers for autism spectrum disorder? And this is something of interest. Ramirez-Salas paper that was published in Molecular Psychiatry in 2021 dives deeper into this space. And what we noticed is the mechanism seems to be autoimmune destruction of critical proteins in the developing mind of the baby. So when we think of that, we say, okay, well, we understand autoimmune disease now. Antibodies floating around in the circulation for one reason or another have developed in response to either toxin exposure, nutritional changes. Something happened that induced autoimmune antibody production from the immune system. And those antibodies, if targeted against certain tissue types, will start to destroy the function of those tissues over time. What did they find in the Ramirez-Salas study? Well, they identified proteins, collapse response mediators, proteins 1 and 2, otherwise known as CRMP1 and CRMP2, guanine deaminase, GDA, lactate dehydrogenase A and B, stress-induced phosphoprotein 1, otherwise known as STIP1, and neuron-specific elonase and Y-box protein 1, so NSE and Y-box. Okay, so these are names of proteins that most of us never know or hear about. Personally, these are things that I don't even think about on a day-to-day basis ever. So then the question is, if they've identified autoantibodies against these proteins, is there mechanistic plausibility to the reality these proteins are causing disease? And the answer is yes. What are these proteins' functions? Let's just look at CRMP, for example. Collapse and response mediator protein is involved in neuronal network formations. Antibodies against CRMP can alter neuronal formation and network activity, leading to dysfunctional brain activity that is seen in autism. So we can say now there's multiple pathways to potentially developing a neurodiversity or neurobehavioral difference that we wouldn't otherwise expect in historical terms. But now we're seeing, again, epidemiology says 1 in 5,000, now down to 1 in 30 plus or less. Big deal. Why is this happening? Well, now we have multiple possibilities. Immune activation, mom gets pregnant. Immune autoantibodies developed in mom attacking child, right? And then we have nutrient changes. And now we're having data looking at toxin exposure. We'll get into that. And there's piece after piece after piece of emerging data sets saying that there are multiple ways to disrupt the system. And I don't think it's a reductionist approach of one thing. The probability is it's multiple things hitting on multiple pieces at the same time, sort of like a dike with multiple holes in it. You can put your finger in one hole, but the water's still coming out. So if you fix one problem, but these other ones are there, we may still end up down that primrose path in the direction we don't want to go to. How does the brain develop? This is an important slide to understand. So if you look at this picture here, you start to get an understanding of what's going on. Well, in the first 36 weeks of life, you see right out of the gate, closure of the neural tube, proliferation of neurons, neuronal migration, differentiation and growth into axons and dendrites, production of synapses and neurotransmitters, development of intercellular signaling systems, right? This all happens in that first 24 weeks of the first 2 trimesters. But then in the 3rd trimester comes the critical phase, glial cells and myelination. So this is where the immune system becomes heavily involved. Development of the hippocampus for memory function. And then we see postnatally, you start to develop transient cortical structure replaced by a different cortical structure, right? And then you have rapid prefrontal development. And on it goes into this beautiful brain that we know of as how we become ourselves, or in the beginning, a child's phase. But if you think about this 0 to 36 weeks up to 40 weeks of gestation, there's a lot going on here that is super critical, from the cell division of the sperm meets the egg all the way to this complex organism that we are as humans. And are there pieces along the pathway of the continuum of this development, of this neurodevelopment, that are sent sideways by different issues that we sort of were touching on in the beginning? Autoantibodies, nutritional inputs, potentially toxin exposures, or is it neurometabolic changes, or all of the above? Well, we know during brain and neurocognitive development, we need macro and micronutrients all along that continuum. right? We also know now that the microbiota is very important here and very, very fundamentally involved in metabolism and the transfer of macro and micronutrients throughout the body. And these microbiota are involved in all levels of how we immunologically respond to the outside world. And we'll get into this a little bit more. But just suffice it to say, if we look at the slide where we get a gross breakdown of lipids, vitamins, amino acids, and minerals, you look at the context of how these macro and micronutrients function. Lipids, super important. Long-chain fats. We're hearing more and more data about the effect of omega-3 fatty acids, specifically docosahexaenoic acid and icosapentaenoic acid, involved in cellular activities in the brain. Fish oil, specifically, as the main source of omega-3s that's critical, developing pro-resolving lipid mediators to decrease inflammatory responses. What's happening in society? Much less omega-3 exposure. We have these lab tests, omega-3 index, we can look at to see if there's something awry here. But just suffice to say, we know neurologically we need these things, right? Polar lipids, right? They're heavily involved in brain and neural development. And what happens when you're missing omega-3 fats, these long-chain polyunsaturated fatty acids, or you're missing the polar lipids? Well, we know that deficiencies lead to change in cognitive ability, change in memory, change in neuronal plasticity, change in executive function, language. We have attention problems. What do all these sound like? These are all pieces of continuums of neurobehavioral disorders that we see of as ADHD, autism spectrum disorder, behavioral problems, all the above. Vitamins. We look at some critical ones here. Vitamin A, right? Involved in relational memory. Dysfunction equals cognitive function decline. B12 and folate, huge, especially if you have defects in MTHFR, MTRR, catechol-methyltransferase, different single nucleotide polymorphisms involved in the shuttling and utilization of B vitamins, which are critically involved in the Krebs cycle and in different cycles throughout our body. We need these cofactors to allow ourselves, as Bonnie Kaplan says, to develop the brain the way we want to. Basically, knowing that these micronutrients are super important. B12 and folate are involved in cell division, myelination, synaptogenesis, neurotransmitter synthesis, and hippocampal development. Lack of these two could seriously impair the growing mind. Vitamin D, been talked about ad nauseum. Everybody knows how important vitamin D is for bone health. But the bigger piece for me is the insufficiency state that's involved in immunologic health, specifically immune tolerance. And we'll get into that a little bit. Different minerals, iron, zinc, iodine, super critically important. And again, you can look at the slide to see a little more in detail. But again, each different mineral is involved as a cofactor and function of major neurotransmission, synaptogenesis, neural transmission of neuroplasticity, all the above. And then amino acids, of course, we need those to build our neurotransmitters, to build different functional pieces of the continuum that we call the nervous system. Probiotics and prebiotics, right? We know this now. We'll get into this a little bit later again. But how about human milk oligosaccharides, recently identified in the last couple decades? How important they are in breast milk in order to help our gut bugs grow appropriately, and that the switch to formula has dramatically reduced this functional ability. All of these things are tied together. There is no separating, reducing this down to one single thing. So when we say, okay, we added DHA back into formula and that's going to fix everything, not a chance. Do we need to add it back? Yes. Is it the single answer to all these problems? Absolutely not. Another way of looking at this slide here, looking at neurogenesis, synaptogenesis, myelination, synaptic pruning, shows this continuum over time and where each piece is super important. And for me, I really am honing in on that first 3 trimesters of life where the human is inside mom, because that's where these critical things are playing out. And I think in society, we're seeing a big difference now, where the maternal health piece has to be at the forefront of understanding why things are going sideways. Again, if the data is to believe that the Duke study is showing that they're able to predict these children at 30 days of age based on certain metrics, then we need to start taking this very seriously. The upstream place to be looking is primarily in utero. Not to say it couldn't be postnatally, not saying that at all. But I think the predominance of data in my mind, again, from what I've seen in 25 years of private pediatric practices, I think the vast majority of the insults are occurring in utero, not post-utero, okay? What are the big players that pull here? Infections, as we talked about, chronic inflammation driven by nutritional inputs, micronutrient deficiencies, environmental stresses. We're going to look at each one of these individually. Timing is always the key. The developing brain is incredibly vulnerable in the early days of life, especially if we find out that you have single nucleotide polymorphisms in the ability to deal with toxins, deal with nutritional movements. Do you have solute carriers that are not functioning as well as they should be? The 8-cylinder engine is now running at 4 cylinders, so you need more of this nutrient, choline, zinc. There's all kinds of stuff here to look at, right? But for the sake of this discussion, we just know timing is important. And we're going to get a little bit about circadian biology. where the timing of how we receive something, either is in the morning or the evening, makes a huge difference now. Epigenetically, we understand these things. The antecedents and triggers, timing makes a big difference. For example, the risk of iron deficiency varies with pediatric age. Peak incidences are seen in the fetal newborn period, 6 to 24 months of age. Big difference, right? But oh, by the way, what about iron deficiency in utero? We know there's data there. And so we're always trying to make sure that people take their prenatal vitamins with iron to make sure they're keeping their sufficiency states up for those cofactors. But I don't think as a medical body now, the obstetrical world is focusing heavily on making sure the macronutrient piece of the micronutrient delivery system is where it should be. Prenatal vitamin may be a benefit, but it may not be the substitute for a perfect, healthful diet, right? So I don't think we can just rest our laurels on saying, hey, here you're taking your prenatal vitamin, don't worry about the rest, right? No, I think we need to be very careful that everyone has a different ability. We are all ends of one as humans, and differential abilities to absorb the food, take it in, and we'll get into this in a slide, but that's really critically important. And then this doesn't even take into account socioeconomic determinants of health, right? If somebody's living in a more impoverished environment where their food quality is worse, the prenatal vitamin is going to be super important. Are they taking it? And then, are we trying as a society to give these folks who live in environments where they're put upon in the ability to get high quality nourishment, are we giving them support systems to help them get to the level they need to in order to have a child that's super healthy? I think we need to work on these things heavily. The cofactor issue doesn't just deal with an antenatal piece, but also deals with a neonatal piece. We need to make sure children are being fed appropriately postnatally to get the appropriate nutrients for the brain to continue growing postnatally. And the volume needs to be coupled to the amount that the child in specific would need, right? And then the best way to do that is through food, and then some more food. All nourishing based, please. RDA is not the metric I would think we should be chasing. RDA is just a metric that says you have enough not to be in the deficiency state. Personally, I'm not looking to worry about the deficiency state. I'm worried about optimization. Rickets will be gone if I make sure you have immune function health done. I know your vitamin D is to the point you're so sufficient for immune health. Rickets is a non-issue. But if we're just worried about rickets, then you may not have immune solvency. So that's the key with the RDA. It's just to deal with deficiency states, not the answer for what we're looking for. Now, if we look at this slide, this is a great one. I love this one. This is the one that shows you how complex this system is, right? We talk about Krebs cycle. We talk about the methylation cycle, how everything spins around, and all of these cofactors are important for this system to work appropriately. But if we look at that, what do we get out of this circular system of chemical development, right? We have neurotransmitters that come out of this, right? We develop all kinds of different pieces of what we call the brain pie, serotonin, dopamine. They come out of that side. We have tryptophan and tyrosine. Right there is your methyl tetrahydrofolate, your MTRR piece. Then you look at your methionine to homocysteine side. We see all of these things, these words we hear about all the time. Elevated homocysteine is an increased risk of cardiovascular disease. Oh, it's also increased risk of other problems, right? If you have hyperhomocystinuria, you can see the downstream problems with ocular lens development, different problems with brain development. So every ounce of this slide, the biochemistry of it's super important for us to start to understand what do we need long-term from a micro and macronutrient perspective to have the brain develop appropriately, right? And that's the key. And then you can also see on here, if you have a SNP in certain parts of the system, the ability to clean toxins out if you don't have the effective function of glutathione, right? And so we're not going to belabor this anymore. Just know it's a complex system, can't be reduced to one problem, multifactorial, but we need to pay attention. Nutrients are the key as far as I'm concerned. I think the most important thing we should be pulling on as far as a lever of health for the growing brain is macro and micronutrients. Maternal health is critically related to how much micro and macronutrient intake we're putting into the system. If you are deficient in micronutrients, you will have decreased development of the neuronal pathways, neuronal metabolic function, myelination, trafficking, and you will likely induce glial abnormalities, which induce mitochondrial damage and lead to the hallmarks of all the diseases we talk about of neurodiversity. Macronutrient deficiency is a little bit more structurally involved. That would be decreased volume of the brain cell number would go down, synaptic number would go down, altered neurotransmitter. That doesn't tend to be as true now because we are not calorically deficient almost ever. The difference with calories now is they're pretty pro-inflammatory, so they're causing more problems on a different level than we are actually macronutrient deficient. That poverty cycle of deficiency from a macronutrient perspective is not there. Now it's a poor quality macronutrient perspective. Let's focus on iron here. I think when we look at iron as a micronutrient, we know it's involved in many, many different pathways in the brain. right? Involved in neurotransmission. It's involved in the entire mitochondrial pathway for cytochrome activity in the electron transport chain, which is how you make all the ATP for all the neural processes, right? And we know acetylcholine is involved here, right? So all this stuff comes to play. Well, how do you need to get iron into the system to the volume you want? Intake and absorption, right? Key. You got to get it in. So if you're not eating it, the question is, are you absorbing it? Well, we know now that people who have gut dysbiosis or intestinal inflammation, which is the vast majority of Americans right now, you're not absorbing stuff as well from the inflammatory perspective. What about the ability to transfer it? Do you have effective transferrin proteins? Are you actually taking it around the body? Or do you have a polymorphism in your ability to carry your iron around? And then what about receptor binding? Is it able to trigger the receptor, the transferrin receptor to grab it? Does it do a good job there? Does it have the ability to get stored in the ferritin cages appropriately? Is hepcidin the main protein involved in up and down regulation of the activity of iron movement? Is that working appropriately in your liver primarily, right? And all this is in concert to understand how iron is supposed to transport around the whole body. And I think it's super fascinating to think about now in this modern society. For most of humanity, we dealt with infectious disease burdens, right? If you think about this, up until the recent past, last 150 years, we mostly dealt with infection as a major source of death. Now, with the advent of antibiotics, sanitation, vaccines, quality nutrition, that really went sideways. We don't see infectious disease burdens as the major reason people are dying. Now, are people still getting infections? Yes, ear infections, pneumonia. Yeah, we're seeing that stuff. But we're not seeing it like we used to, where people are dropping off the planet from 0 to 5 in a high volume, where people had to have 9 kids in order to keep 5. That's gone, right? So we know our system was set up so that, for example, if you had an infectious disease burden, one thing the body knew very well was that iron is utilized by the bacteria and viruses. So what would the body want to do if you're infected? Well, it would say, okay, I don't want to have iron floating around for the virus and bacteria to replicate, so I'm going to hide them. And where would I hide them? It'd put them in ferritin cages, right? And so your ferritin level goes up, which is a sign of acute phase reactant, which is how I learned in medical school. Ferritin elevation, acute phase reactant, right? In general. Now, you could have hemochromatosis, but we're not talking about that. We're talking about ferritin elevation purely from inflammation burden. Well, that's interesting. So your ferritin goes up because your body's trying to hide it from the bacteria and the virus. That's super cool. There's a reason behind the why. Now we go fast forward and we say, okay, well, why is ferritin being hidden now? A lot of times it's not infectious, unless it's potentially systemic dysbiosis going on, so your gut's leaking and so much LPS possible. But more likely, it's related to autoimmune inflammation or some other form of inflammatory disease burden that is causing the same process to happen. That's super interesting. So now we know, okay, you need to take in the iron, absorb it, transfer it, get it bound, and utilize it. But oh, by the way, that could all get sent sideways by a lot of these antecedent problems that we're talking about that are completely understood in functional medicine from the perspective of the global picture of intestinal dysbiosis, food-derived inflammation, chemical-derived inflammation, all things that you hear every time we go into a lecture hall with folks who understand the true reality behind what's going on inside the system. So this is just iron. This same picture would go for zinc. This same picture would go for selenium, choline, B6, everything. So again, not one piece, big pie, one part of the project that we need to understand, but you need to understand the whole project to really get it under the belt of trying to change, right? Well, this slide we look at, and I think it's a great one to look at because, again, it's another way of seeing it. So what does the neuron need for function, right? To grow, it needs protein, carbohydrates, fats, iron, copper, zinc, iodine, vitamin A, D, B6, and C. That's just the neuron. What about oligodendrocytes? Protein, carbohydrates, iron, iodine, selenium, zinc, vitamin B6, B12. What about the astrocytes? Protein, iron, iodine, copper, zinc, selenium, choline, B6, D. You see a repeated pattern here. But there's differences here. So if you're really good in some but not in the others, you may not get to all of the functional realities of getting the system optimized. So you have to want to look at each piece of it. Are we getting in the micronutrients and macronutrients for the effective anatomy to get it to where it needs to by the end of pregnancy, and then from there on at least till age 3. Super, super important. Here's one study by Friedman et al. Higher gestational choline levels protect fetal brain development and maternal infection, right? So this is a couple articles for you to look at. Journal of Pediatrics, right? They said, all right, does choline, right, which is a major donor of choline, right? But choline also is a cofactor in many other functions. right? Is choline a part of the fetal brain development cycle? And the answer is clearly yes. What about this Dr. Zeisel's work? He's here in North Carolina where I'm at. And he looked at this and says, is choline an essential nutrient for public health? Was it? And the answer, of course, was yes. And here's a nice picture looking at what is choline used for. Well, it goes back and forth between acetylcholine, phosphatidylcholine, which is involved in all kinds of cell surface molecular function. Well, it also can get down into methionine, right? And it goes down into homocysteine. This is where we get into methyl donors. It is involved in the folic acid pathway. And you see this, oh my gosh, this nutrient, this single nutrient choline is seriously involved in massive pieces of what we would consider the neurodevelopmental pathways. So it's no surprise that the studies are showing how important it is. Where we get choline from? Well, eggs are a great source of choline. But a lot of folks don't consume the volumes needed for function, especially when pregnant. Big deal. A lot of the prenatal vitamins don't even have choline in it. So we should be really thinking about that one. What about zinc? What do we need zinc for? Right? Well, we need zinc for biological functions like oxidation reduction functions. We need it for transfer functions of different micronutrients, right? We need it in the structure of different DNA ligases for DNA repair. Zinc finger is involved in DNA transcription, DNA repair. What are you hearing there over and over again? DNA. What is DNA involved in? Growing the body. Now I'd like to shift into metabolic states for mom. How is the immunometabolism of the body shifted in the past 50 years or more? And is it pro-inflammatory? What are the upstream effectors of maternal immune activity? What drives the increased pro-inflammatory maternal child immune cell polarity that we're seeing in study, the dyad of change, right? The dyad of the mom and child. Something's happening to mom that's affecting child, that's leading to this neurobehavioral change from 1 in 5,000 for autism to 1 in 38. Well, for me, I think we really need to look at the immune system because for me, everything starts and finishes with the immune health of the human, right? Well, we know in pregnancy, one of the most important critical pieces of pregnancy is that in order to survive, the genomic understanding that half of the gene of the child, the gene pool of the child comes from dad, half of it comes from mom. Well, knowing that, what do you need to do? Well, you can't allow the immune system to attack dad's foreign DNA because if you do, it's going to abort the child. And this actually happens in Turner syndrome and other dysfunctional realities where the T cell system is polarized towards a Th1 state in order to abort the child. But in order to avoid this from happening, because we know that mom has a child in her that's half dad's gene pool, and that would be foreign to her, she would have to then shift her genomic structure of T cell polarity. So epigenetically, or however it fully functions and occurs, the T cell programming shifts so that Th2 becomes more the dominant structure. And Th2, if you remember, is the one that is primarily set up to kill parasites, historically. Th1 is generally set up to kill intracellular pathogens. And Th1 is the one that's, going to help turn on cytotoxic T cells. That's going to sort of wipe out pathogens, especially if they're inside your cell. Well, oh, by the way, that would be the one that would wipe out a growing baby. So we know now, if you can look at the slide, non-pregnancy state, you look at all of mom's regulatory T cells and B cells, they're all sort of across this midline pattern. But then all of a sudden pregnancy happens, you see a big shift. B regulatory cells, Treg cells, they start to increase. Regulator cells, again, so the immune system's police to keep everything controlled and not out of control, right? But what happens? Oh, the Th2 increases. Okay, that makes sense. That's the one that's not going to go after and kill the baby. Phagocytic systems, NK cells, the natural killer cells, which are going after circulating cancer cells and circulating pathogens, right? Th1 responses, again, that gets downregulated. Super interesting. Okay, so this is the norm. So hold on to that memory when we look at some further slides. This one, Zavodska, great study. Highly encourage you to pull it. This is a review of the evidence, proposed mechanisms and implications for treatment related to the role of maternal activation, immunity-wise, in the pathogenesis of autism. This was in the International Journal of Molecular Science in 2021. Pull this article. Highly recommend it. Really fascinating. So again, infected-induced maternal activation syndrome, right? So mom gets sick. This was that earlier study where we've seen this with the flu, but earlier I showed it with COVID. Many different viruses can induce inflammatory reactions inside the human mother that actually affect the child. And here we see it. Pro-inflammatory cytokines get turned on by the infection, TNF-alpha, IL-6, IL-1 beta. They crank up Th17 cells, which we know of as the extracellular bacterial fighting cells, but oh, by the way, are also the mediators of autoimmune disease. IL-17A cranks up. This crosses into the placenta. Activation of resident immune cells inside the placenta and inside the baby. Oh, by the way, if those are glial cells, now you start to have oxidative stress and damage in the local microenvironment of the brain, mitochondrial dysfunction, microglia start to kill and destroy local tissue. The oxidative stress is a singular problem, but the whole thing in concert is related to the development of neurodiverse problems, right? How about from a different perspective, maternal immune activation as it's related to gestational diabetes, which we know is related to a myriad of upstream lifestyle choices that drive immunometabolic dysregulation. As we stated in the beginning, a mother who is overweight by the obese classification and diabetic has a 4x risk of neurodiverse changes in their child, right? Gestational diabetes, in my mind, is problematical as well. When does it kick in during the pregnancy? That's the question, how much it's related to dysfunctional problems in the child as they go on. Right, but look at the immune polarity changes. In normal, as we saw before, you should see increase in Tregs, increase in Th2, increase in monocytes, right? Nothing major is going in the wrong direction. Then you slip over to the other side, the diabetic side, right? And I think, again, this would be easily shown if somebody was truly diabetic. But this study was just looking specific at gestational diabetes. And what do they find? Neutrophil overactivation, not good if you're making neutrophil extracellular traps. The nets will do some major damage. NK cell activity is cytotoxic in type, so that's not good. Monocyte activation, not good because that's going to increase your ability to make autoimmune antibodies against tissue presentation antigens, right? Th1 and Th17 response, up, right? Th1, intracellular killing capacity, Th17, autoimmune production and extracellular killing, right? So these are immune polarities we don't want to see in pregnancy. Cytotoxic T cells, B cells, all of the stuff gets upregulated in a way that we don't want. Macrophages come in, they get activated. This is not good. So now we start to say, okay, well, this is starting to make sense. If mom's immune system is upregulating in all the ways that we saw in the system of inflammation driving, maybe this is what's really deriving the metabolic changes inside the brain of the newborn while they're inside the body of mom that leading to the phenotype that we see of as autism spectrum disorder. I believe it to be so. What about fructose? And this is a whole nother aside. I'd love to do an entire different talk on this. If you ever have a chance, I interviewed Rick Johnson, who has done some amazing work in this space for the podcast that I do, Dr. M's Wounded Children First podcast. And we talked about fructose. And I'm going to do a little bit of a deeper dive here, but not the full Monty. Fructose, when I was in medical school, was essentially a sugar molecule that we knew was part of the disaccharide we call sucrose. Right? So, okay, you got your glucose and your fructose, but fructose is the part that's super tasty. It's a beautiful, sweet stuff, right? Glucose doesn't really taste like much. So if you actually take an IV bag of glucose and pour it in your mouth, it doesn't really taste sweet. Fructose is the sweet part. And evolutionarily, we never truly understood why that was the case, but now we really do, right? So Rick's work, I think, is super fascinating. He found out in his book, Nature Wants Us to Be Fat, that fructose, when it's metabolized, goes through a process of becoming uric acid. And when it becomes uric acid, it actually turns on a whole cassette of genes inside the liver and in the body that says, store fat in the liver, store fat in the periphery, increase blood pressure, and forage for more food. What does that sound like? Sounds like metabolic syndrome. Well, if that's the case, what happens if we remove fructose from the diet? Well, he did that study and found that, yes, that did help, but it wasn't the whole story because it turns out glucose polymers that we call starch in plants, glycogen in humans in the muscle, glucose polymers have a reverse engineering pathway to go all the way back to fructose via this thing called the polyol pathway, where fructose comes from glucose via sorbitol, and then that you're off to the race again to make uric acid. So it actually turns out that in order to control the volume of uric acid in the body, we get to why that's important in a second, you need to control actually your glucose polymers, which is your starches, your white foods, and your sugars, right? Specifically your fructose ingestion by high fructose corn syrup or any of those players. Now let's get into the understanding of that as it relates to pregnancy, which I believe, again, is one of the main reasons we're seeing autism spectrum disorder increase in this generation of high sugar intake in children and their pregnant mothers. Maternal high fructose ingestion associated with anxiety and inflammation and so much more has been studied and looked at when it comes to pregnancy. But in Rick Johnson's work, he found that the placenta, as it's growing in the first 8 to 12 weeks, is a glycolytic event, which means it's not using aerobic respiration. It's primarily using anaerobic respiration where you take your glucose and you break it down into lactic acid, and that's how the system gets the ATP it needs to at 2 ATPs per usage of the glucose molecule, and you're building, right? This is normal. Somewhere around 8 to 12 weeks, the spiral artery starts to invade the placenta, oxygen comes in, and you switch to aerobic respiration. You make a lot more ATP this way, and beautiful. Now the child's developing, everything's lovely. Well, in his work, he found that women who are consuming larger volumes of fructose actually had changes that were relatively dysfunctional as it comes to endothelial dysfunction, right? So if you look at the slide there, we see in this picture that the hypoxic placenta will, use HIF1 alpha, hypoxia-induced factor 1 alpha, which actually is a main driver of NF-kappa B. That's another aside. That via fructokinase, it'll increase uric acid, which increases vasopressin, which then will increase endothelial dysfunction, which impairs trophoblastic invasion, immature spiral artery invasion, and then you're back to hypoxic placenta that never stops. In case you see this sort of pattern, the hypoxia doesn't go away, you stay glycolytic, you increase endothelial damage of the placenta, which leads to preeclampsia. And in the fetus, we see associated with this intrauterine growth retardation, metabolic syndrome, hyperglycemia, and reduced number of litters for mom, right? And what do we know from vasopressin, right? Well, vasopressin, you will have elevations in blood pressure. Ooh, that sounds like preeclampsia and eclampsia. What about fatty liver, proteinuria, increased, insulin resistance? These are all the things that we talk about from an OB perspective, but we now have some pretty good evidence that this is being driven by uric acid, which is completely being driven by fructose and glucose starch polymer. That's sinking in. That's to me, this is super, super important, because I think if we can pull back on these levers, these massive levers of metabolism changes in humans, we may have a chance to unwind some of these antecedent risk factors for dysfunction. And again, all based on immunologic activity, right? When we look at this, what's really happening with uric acid is activation of NLRP3 inflammasomes. NLRP3 inflammasomes are major pyrin-mediated, so fireballs of inflammation, right? So that's where a lot of this stuff is actually happening at the cellular level. But the driver of that is uric acid. What's the driver of that? The pathways involved in the degradation of fructose to uric acid. So let's switch to breast milk. All right, breast milk is the complete food for babies. Well, what about if you're a mom who has gestational diabetes and you're breastfeeding? Well, we found out that there's a change in the bioactive compounds present in the colostrum that may affect development of the newborn's immune system. In this one study by Desena-Avalle in the Journal of Frontiers in Immunology in 2022, we see that the gestational diabetes mothers had higher secretion of cytokines and chemokines in the colostrum. Again, this is the first 2 days to 3 days of breastfeeding, when the concentrations were higher of gamma interferon, IL-6, and IL-15, and lower concentration of IL-1RA, right? This is super interesting because those are pro-inflammatory. And when they looked at growth factors, they identified decreased concentrations of GM-CSF in the colostrum of diabetic mothers. So this all leads to, again, the possibility, this is another piece of the hole in the dike that's leaking water, dysfunctional breast milk. And again, now we can go back and say to our slide of the continuum of when things are happening, okay, that's a big piece of this. The nutritional immune polarity piece, the infection possibility, the nutritional possibilities, the fructose, the different pieces of this pie. We're pulling on some of those levers right now. We're going to get a little deeper, but I just wanted to go a little deeper on, at that point, just on the immunology. Now let's shift gears to the exposome. All right, everyone knows what the environmental exposome. Basically, it's anything that attacks your body or attaches to your body from the outside world. This could be stress, living with somebody who's very angry. This could be a child receiving adverse childhood events when young and that carries to when they're older and pregnant. This could be food. This could be toxin. This could be inhaled poor air as a toxin. This could be antibiotic exposure. I mean, anything essentially could do this. Why do we care about this? Well, the exposome is heavily related to immune activation. And here we want immune activation in a way that's solvent, not pro-inflammatory, right? So I look at this exposome as the key. We want to be exposed to things from the outside world. We get into a deeper dive in immunology, which we won't go deep today, but we have these pattern recognition receptors, PAMPs and DAMPs, right? PAMPs are the pathogen-associated molecular pattern receptors, and the DAMPs are damage-associated molecular pattern receptors, right? And these suckers are out there to say, okay, outside world, I see you, Mr. EBV virus, Epstein-Barr virus. I'm going to grab you, taste you. I'm going to send you down the primrose path of lymph node, develop B cells and T cells against you, so that when you come back, I'm going to keep you in check so you don't cause trouble. The DAMPs are there to say, okay, local cellular damage happened. We just saw some major cells get blown up. Now we see floating around us some DNA and some ribonucleic protein and some uric acid, and they're actually major triggers for the immune system and inflammasomes to say, okay, something's going on here, let's come in and burn that up. Normally this happens in a great way. You burn it up, everything gets killed, the system cleans all that debris up and says, okay, let's fix everything, right? So that's what we're looking for. But if it doesn't go according to plan, then the PAMPs and DAMPs get triggered. They tell local immune cells, hey, we got a major problem here. If we're hyperinflamed, in that case of the diabetic mother, dendritic cells get educated. Maybe a naive T cell now becomes educated in the ability to make an antibody against CRMP, upregulation of T cells. Now you got effector B and T cell lineage against CRMP. That CRMP is involved in neuroplasticity and neurodegeneration. And now you potentially have an antibody like Ramirez-Ellis' work causing damage to the brain of the child. And then if we think about from an autoimmune perspective, if we have those autoimmune antibodies, what's going to make that risk higher? Well, we know this, dysglycemia. So folks who have glycemic issues, diabetics, gestational diabetics, and folks with obesity who are not diabetic yet, but have huge spikes of insulin, they're getting hyperglycemic. Food reactions, so we're looking at here is food allergies and food intolerances. It's not just celiac disease. There's non-celiac gluten sensitivity. There's many food intolerances in my clinic. The number one food intolerance I see in infants is milk protein intolerance to casein protein. Major problem. We'll get into that one day in another lecture. We have detoxification defects, the ability to see toxins, clear them naturally. Children have a harder time clearing toxins, right? So this is a big deal. Hormones. We're finding more and more endocrine disruptors in our environment. How are those hormones affecting mom? Well, studies are showing this, and I'm going to show that in a few slides coming up. The studies are showing that endocrine disruptors are increasing the risk of poor quality pregnancies. What about insomnia, poor sleep? We need to talk about that. So all that can show in this wonderful slide by Dr. Yannick that will induce inflammation. Well, that increases the risk for autoimmunity, epitope spread, which could be more autoimmunity, and then this could all be tied into dysbiosis and infection. All of it said, when you look at it at the end, autoimmune flares can get worse. How about cell reports, right? So there's a journal article that I thought was fascinating, from Dr. Tan et al., and they looked at this piece of what I call tolerance. And so for me, immune solvency, I keep using this word over and over again. I think the critical piece of immune solvency is we need to understand tolerance. The immune system was set up for us to see the outside world in a way that is natural but protective, i.e., that means that we see a foreign invader as a macrobiotic creature like a Ascaris worm to a bacteria like strep pneumo. The immune system needs to see it, recognize it, develop a good antibody response to it after the T cells do the initial response of killing, and then we need to be protected against it in the future. Tolerance is the part of the system that says, one, after that infection has been received by our body and dealt with by our body, we don't keep staying inflamed. The army goes into stand-down mode or into quiescent phase. That's tolerance. And oh, by the way, if it sees a peanut in that process of getting all churned up in the immune function of the gut, it doesn't react to the peanut in a way that we say is allergy, let's say. That to me is what I think of when I think of immune tolerance. Dr. Tan's article here said, okay, well, what drives Treg activity? What drives the ability to have secretory IgA and normal function of our gut immune system and the circulatory Treg system throughout the whole body? Well, a couple things are known. Vitamin D induces FOXP3, which is involved in T regulator activity. Vitamin A, fiber, right? Fiber feeds the gut bugs. Well, it turns out that that's highly associated with improved immune tolerance. So here are three levers to pull on. Making sure you have adequate vitamin D, adequate vitamin A, and adequate fiber. What's the best source of fiber for a baby right out of the gate? Human milk oligosaccharides as breast milk, with over 220 human milk oligosaccharides that feed the gut microbiome that induce tolerance. It is a massive crime to me, and I don't mean this offensively to anyone who bottle fed their child. To me, it's a massive crime against a child. If you can breastfeed and you can give them the most beautiful fiber source on the planet, human milk oligosaccharides, that you don't. And again, this is not meant to be a judgment. This is meant to be scientifically stated in such that I would love to see the word get out there that formula is not breast milk for many reasons, but one of the biggest ones is the induction of natural immune tolerance. And again, I am saying this loud and clear. I do not want to offend anyone who did not breastfeed their child. I just feel so strongly about breastfeeding should be the only choice if you can. That's all I'll say about that. Let's shift gears. Epigenetics. I think the future of our immunometabolic understanding is going to come through nutritional understandings of how food affects the immune system and epigenetic understandings about what else is driving the epigenetic changes that are turning on the proteins that are leading us to be who we are. If you've never heard me speak before, the Goody Mouse model is the one I like to use. Randy Journal, good friend of mine, did an amazing study back in 2000 where he took a mouse that was called the agouti mouse. And if you do nothing to this mouse, it'll be yellow, prone to diabetes, prone to obesity, and prone to cancer if you take mom and dad mouse, put them together, and they keep producing offspring. His research lab with Rob Waterland, who is a doctoral student, who was saying, okay, I come from a nutritional background. As they were looking at this mouse, they said, I wonder what would happen if we gave food as methyl donors, right? Carbon atom with 3 hydrogens that has the ability to stick on the DNA. I wonder what would happen to those sticky marks if we fed food when they're pregnant. This is where it gets unbelievably mind-blowing. When I heard this the first time, I almost fell out of my chair. They took the pregnant dam and gave her methyl donors as betaine, choline, B12, folic acid, or folate to be more specific. And these came through food again. And they gave the mom, while she was a pregnant mouse, these supplements. And lo and behold, the baby mice came out skinny, not prone to diabetes, not prone to cancer, and yellow coat color was changed to a brown coat color. And when they looked at it, Dr. Jerdal's group found that what was happening was that methyl donor, that carbon atom with 3 hydrogen, went upstream of the transcription region for the protein that is the agouti protein and silenced it. This was the first time we realized that Darwin was not completely correct. That Lamarck, actually, Jean-Baptiste Lamarck, had understood that in a lifetime of a human or an animal, you will actually alter your function through experience. Darwin said change in the DNA only occurs via death and what we called natural selection. But he was right that hardwired DNA change is only going to change based on if somebody dies out, then the other gene will proliferate. We could talk about that with the uricase mutation that Dr. Johnson's work is, but we'll skip that for now and just understand that he was right, but he was incomplete. Jean-Baptiste Lamarck said, I think in the lifetime we're going to change. And Randy Jerdal's work proved that. If you give methyl donors these carbon atoms with 3 hydrogen, they will put a sticky note that says, don't read me to this protein transcription region on the DNA. Didn't alter the DNA, just changed how it was read, and the animals came out completely healthy. That was not the end of the story. His next postdoctoral fellow was Dana Dolinoy. Dana Dolinoy said, okay, I'm really curious as to what happened. She came from a toxicological background. And she said, what happens if we add BPA to that same story? Keep doing the same story, but add BPA, a known hypomethylator. BPA is bisphenol A. It's a plasticized within hard plastics, and unfortunately, we used to give this to babies. God, that's insane to think of. We used to give this to babies. The BPA was given in aliquots to see what would happen, and oh, by the way, eventually, it removed the sticky note. It removed the carbon atom with 3 hydrogens. Next thing you know, the babies, the offspring of the mouse, came out yellow, prone to diabetes, prone to cancer, and prone to obesity. So now we see the most important piece of an understanding that you have the ability to do benefit with food and the ability to do harm with chemical. And this is a repeated theme, I think, throughout the history of humanity that we need to recognize. Like, we're not doing enough as a group of physicians to understand how much we're toxifying our environment by allowing chemical companies to do what they're doing. Now, this is a translational model, but we know that this is happening in all species, right? And it's a big mess. So this is the primer. This is the piece of understanding. This is the way I want it to be said. So you really start to get a big picture of, holy cow, when I start talking about food, how important food is, here's a big piece of the why. When I start talking about chemicals and how chemicals are involved in this, here's a big piece of the why. Epigenetics is not just a prenatal and perinatal event. It's throughout your entire life. For the subject of this growing brains topic, it is primarily a prenatal event. Maternal metabolism, we know that's involved. Severe infections, we know that's involved. Toxin exposure, we just talked about that with BPA. Oxidative stress, we know that's involved, especially in the brain with microglial activation in an infection state, right? Maternal nutrition, right? Just like I said, maternal metabolism, that's tied to maternal nutrition. Placental dysfunction, fructose driving that, phthalates driving that. I mean, we can get all in many, many different topics of this, but this is a big deal. Nobody's ever said I love toxins. That's just the simplest answer on the earth. I don't understand. I'll never get political on these talks, but let me just say this. I would love to see candidates for government sticking to 3 things primarily: clean water, clean food, and clean air. When we have those 3 things fixed first, then let's talk about everything else. Until we do that, we really don't have anything. Because if we don't have health in long term, all the rest of the stuff is smoke and mirrors. So I would love to see toxins as a focus of removal from our world, right? You see in Detroit, lead exposure. You see in Chemours dumping chemicals into the Cape Fear River in North Carolina. I mean, you see all these different things. You can read them all over the place. The big train derailment with the very dangerous chemical that they released in Palestine. I think it was Ohio, right? I mean, over and over again. That's getting into the groundwater. You can pretty much guarantee it. Terrible area for potential future living. These things need to be dealt with. Timing of toxin exposure, huge, right? 10 American study by EWG, Ken Cook's group, the Environmental Working Group. That study showed that when they looked at the cord blood of 10 babies, they found that they were exposed to over 270 chemicals in volumes high enough to potentially cause disease. 400, and I think it was 13 tested. I don't remember the exact numbers. This is a big deal. This is cord blood. So if we're talking epigenetically, massive. How much do we know these are not causing it? It's so hard to pull on that lever and say this is exactly it. But I can tell you what they aren't doing. These chemicals can't be doing anything good. There's a recent study in nanoplastics, nanomaterials in 2023 by Kopats et al., where they showed that nanoplastics are getting into the brain. They're crossing the blood-brain barrier. What's that doing? Well, we know nanoplastics turn on NLRP3 inflammasomes. We do know that in study. So what's that doing? Probably increasing inflammation. Can dudes say anything past that? Nope, but that can't be good. What about recent environmental health studies? Prenatal PM2.5 exposure increases neurodevelopmental risk at 2 years of age. That was a study in the Environmental Health Journal by Morgan in 2023. So just a couple I just pulled recently. There's so many more out there. You can look at this ad nauseum, but that has to be a part of this. We know phthalates are now in the news. Talking about phthalates causing increased risk of preterm delivery, increased risk of pregnancy-induced complications, including IUGR and preeclampsia. Phthalates are like BPA. They're endocrine disruptors. What about caffeine, right? High-dose caffeine turns out to be a problem in pregnancy with early pregnancy losses, with early miscarriages. So you wouldn't want to do a lot of caffeine. Frankly, as far as I'm concerned, all chemicals should be removed from your body before you even decide to get pregnant. And coffee has caffeine in it, and it is a chemical. Soda has caffeine in it. Green tea has caffeine. I would say just avoid all those before you want to get pregnant. And frankly, especially when you do get pregnant. And the problem is you don't know for 4 weeks, maybe 6 weeks that you're pregnant. So you wouldn't want to be taking these things and potentially doing damage at the most critical time when the cells are just first dividing. And then we think about, here's a study in Frontiers in Microbiology by Gamma et al. And it's called Effects of Dietary Pesticides on the Gut Microbiome and Neurodevelopment, right? Again, another one. And what are they looking at here? Glyphosate, 2,4-D, chlorpyrifos. Well, they found it in the water, in the soil, in the foods. They noted that it causes gut dysbiosis. That can't be good. They noted it got into the breastmilk. That can't be good. Right? And when they looked at it, they noted that this was associated, again, not causally, but associated with neurodevelopmental outcomes in children. Okay, so this is a big deal for me again. It's impossible to prove causation because it's too small of a signal in general, but they can't be doing anything good. So why do we want them in there? If we know that we've gone from a 1 in 5,000 risk of autism spectrum disorder to 1 in 20 to 1 in 38, depending on which study you look at. Chemicals need to be out. Maternal diet and child's diet, we just need to improve it. We need the methyl donors. We need B9. We need B12. We need choline. We need betaine. We need these sources, right? And we can get them from so many different foods. Let's switch gears again. How about the microbiome? We know the microbiome is everywhere in the news. There's thousands upon thousands of articles every week on the microbiome. And now the maternal microbiome is in the news constantly with what's happening. We now know that we're seeing a depleted microbiome in women comparatively to ancestral historical pasts, right? What does that do? Well, we know from a systems biology perspective that a rainforest that has diverse species has the best ecosystem of health. It's no different than a human microbiome. If you have diverse species, you have the best immune microbiome of health, right? With bacteria. And we know the bacteria, again, is tied intimately to the immune system. So if you have good quality bacteria in your gut, you have a really great chance of having an ecosystem that is biologically in your favor. And if you don't, well, then you have decreased tolerance, increased immune dysregulation, all things we talked about earlier, autoimmune risk, maternal activation syndrome risk, blah, and on and on. Circuitous, circular reasoning where they feed each other back and forth, back and forth, back and forth. We know that maternal immune activation is related to dysbiosis. So we go all the way back to that earlier slide where we showed maternal activation from SARS-CoV-2 or flu or other bugs is related to dysbiosis. Look at SARS-CoV-2 as far as just general health death rates, right? How many people talked about in the modern news media the main cause of death in the United States from SARS-CoV-2? They all said that it was 4 diseases were associated with 98 every 100 deaths: diabetes, heart disease, hypertension, and obesity. What's the one common theme to those 4 diseases across the board? Food, and then secondarily toxin exposure, right? That will drive both of those diseases. But did anyone talk about that as a way to reduce your risk of dying? Nope. Vaccinate, take masks and wear them, and social distance. 2 of those are not very great for social health, right? Which increases stress in the body, which isn't great for our human selves, right? So ultimately, we want to avoid the foods and the toxins and the poor exercise capability and the other things that are driving immune dysregulation and dysbiosis in the human frame. That's another lecture, again, dysbiosis. There's so many antecedent triggers to dysbiosis, which I think is one of the, again, main reasons we're having immune dysregulation. Dysbiosis, which is, again, is being driven by the main reason we have immune dysregulation. How about the maternal microbiome in and of itself, right? If we know mom has a depleted microbiome, is there a way to enhance that? I say the answer is yes. Lots and lots of fiber, good quality, healthy foods, right? Avoiding the foods that are not good for us, avoiding antibiotics, antacids, and we can go down that road for a long period of time. But we want to enhance the maternal microbiome so that what mom gives to her daughter or son is a healthy microbiome. And then if you are delivered vaginally, you receive that microbiome through the process of being delivered vaginally. But what if you don't get delivered vaginally? What if you end up in a C-section? Well, that's unfortunate. Then you're not going to get that transfer of microbes. Well, maybe we should be doing vaginal microbial transplants. I think we should. I've heard it said way too many times that that's not safe. I think that's absolutely incredibly crazy. And here's an article that was just published in Cell Host and Microbe by Zhu et al. In 2023. The study looked at 68 cesarean-delivered infants who were randomly assigned to receive vaginal microbial transplant or a saline gauze. Adverse events were not significant, different between the two groups. Infant neurodevelopment, as checked by an Ages and Stages questionnaire, ASQ3, at 6 months of age, was significantly higher for the group that received the microbial transfer. Not surprising. This is what nature intended. You come out through the vaginal canal and you get a microbial transfer. Brilliant. This is what it should be. Vaginal microbial transfer also significantly accelerated gut microbiota, the maturation of it, and regulated levels of certain fecal metabolites and metabolic functions, including carbohydrate, energy, and amino acid metabolisms within 40 days after birth. That's super important, again, because now if you are truly fixing the way we metabolize carbohydrates, sugars, and fats through the intestinal microbiome being healthy, you're now going to have immune regulation and normal weight. It's a beautiful thing. It makes complete sense to me. And then overall, the fundamental answer is vaginal microbial transfers are safe, with the exception of maybe if somebody has AIDS, that's active. If somebody has active GBS infection that they know, or if they have active herpes. Okay, so those are ones you know in advance. Those are ones you wouldn't do it to. I'm not sure what else makes sense to me why we would not do this. The bugs matter. Avivo, a probiotic, I think has some of the best data. Dr. Hendrick et al. Did a study in the journal Cell in 2023. Highly encourage you to read it, where they looked at this probiotic, Bifidobacter infantis. It's a subspecies, EBC001. It's one of the only bacteria known that's normal to humans, which we don't have much anymore, to the tune of somewhere around 90% in developed countries don't have it anymore. Is the only one that has all of the enzyme genes available to make, the bacteria can make the enzymes to break down the human milk oligosaccharides so that the short-chain fatty acids and the metabolites that come out feed our intestinal cells of our babies. All right, this is brilliant. And I think we should be talking about maybe Avivo should be given as a probiotic to moms. I have no financial interest with this company, so please take this for what it's worth. I think that we should be talking about this. If it's not Avivo, it's another probiotic, but something that helps enhance the quality and capability of a maternal microbiome that's given to the child so the child does better. Let's switch gears a little bit again. Stress. How is stress related to this process? Well, I think clearly that stress is terrible for the body. Acute stress, totally fine. You get it, you get over it. Sort of the bull's chasing you. Everything shuts down from a parasympathetic perspective. It's all sympathetic overload. You need blood to your heart, blood to your muscles, blood to your brain that says get away, and you run away, and then everything stops and goes back to parasympathetic. What is parasympathetic? Well, we know that's your relaxed state. Digestion, normal breathing pattern, normal heart rate, normal immune activity, all the stuff that we normally need to function on a day-to-day basis is parasympathetically driven. That's our relaxed state. What happens if you're chronically stressed, right? Does your parasympathetic system stop working appropriately? I think the answer is yes, right? Well, we know this now. A lot of the data talking about meditation, prayer, breath work, everything that's being used to help the system work better, it's because it's unwinding what stress is doing to the immune system. Well, what does it really do? Well, stress induces cortisol. And cortisol, we know, is the hormone that increases blood sugar. It increases all the things we need to get away from that bull. Big deal, epinephrine. But what if that stays on all the time? Well, that's hyperglycemia. That's too much epinephrine, which means that's too much stimulating neurotransmitters. Oh, by the way, in the acute phase, NF-kappa B inside the cell is actually turned off, which is super interesting. So inflammation is actually expected to be off during a sympathetic run from the bull event. Well, in the parasympathetic state, the opposite happens. If you are in a relaxed state, NF-kappa B can turn on as needed for inflammation to deal with whatever happened maybe during the acute phase. So this would make sense. But in a chronic state, there is a problem with a receptor that cortisol hits. And there's a cortisol response element that actually becomes somewhat resistant through a dimer that's P50, P51, if I remember correctly, that doesn't now translocate down the way it's supposed to, and NF-kappa B turns on ad nauseum. And if NF-kappa B turns on ad nauseum, it is one of the major pro-inflammatory cytokines inside the human body. So it would not be a surprise that children with adverse childhood events, women who've been abused, men who've been abused from whatever way that would be, physical, sexual, mental, any of those, you might have a dysregulated inflammation system from a dysregulated hypothalamic axis from stress. Something to think about. What about sleep? I think sleep is a main player. Again, another huge lever to pull on. Do we know what sleep does when it comes to immune function? We do. Poor sleep causes downregulation of Th1, upregulation of Th2, downregulation of NK cells and CD8 T cells. B cell antibody production goes down, antigen presentation goes down, inflammatory mediators go up. So in general, sleep deprivation drives big-time inflammation, which unfortunately can lead to neurodegenerative problems, right, as we age. But, oh, by the way, this is also hyperinflammatory problems that could be going on in utero if that's the blood chemical milieu of cytokines going to the child via the placenta. So to me, that's worrisome, right? So if you are sitting there with a sleep-deprived state for a long period of time and you get pregnant, you have a chemical structure inside your bloodstream that's passing through the placenta that is pro-inflammatory and is not good. Sleep is critical. And we know sleep is always related to circadian rhythms, right? We need to talk about that too. But the bottom line is that we need to be very fully in the system of recognizing that the system is meant to go up and down with the sun and the moon. And if we don't go up and down with the sun and the moon, we increase our risk of having dysregulated cellular activity. In this slide, we see short sleep duration, sleep-disordered breathing. So in this case, you would think of sleep-disordered breathing primarily in obese state, would lead to decreased total sleep time and slow-wave sleep. That would cause decreased glucose tolerance and decreased insulin sensitivity, which then would lead to all the factors that we worry about a lot: gestational diabetes, fetal distress, birth complications, poor fetal outcomes, all of which are associated with autism spectrum. And then you see to the left there again, you have the sympathetic nervous system overload with cortisol, inflammation, hypoxia, all in this loop pattern. This is sort of the key. The basics are sleep sufficiency is tied to immune solvency. Sleep sufficiency tied to immune solvency. Circadian rhythms are a part of this. We need to really sort of look at that a little bit more as well, because if we don't understand circadian biology in an appropriate term, we don't really know then if we're sleeping during the day and we think that's okay, in the long run, we may end up with more problems than is good. Circadian in and of itself, the word, comes from the Latin circa diem, or about the day, as opposed to, the opposite, which would be, instead of diurnal, it's nocturnal, which means at night. And to me, since the history of humanity, most everyone followed the sun and the moon because that was your lighting, until natural light was not the only source of light. When we developed the light bulb or candles, even that candles, the lumens coming off a candle's minimal. When we developed this bright natural light, that's when we really started to see that maybe now we're messing with the circadian biology of the human. And it's very clear now in study that circadian biology drives immune activation. And if you look at the studies that discuss when the immune system activates, it primarily activates when the sun comes up. Why? Well, this would make complete sense anthropologically. What would you do when the sun came up? Well, you get your day started, which means you're going to interact with other humans. You could be sick. You're going to interact with your food. So you may be cutting an animal to eat it that morning. You may be out foraging or hunting. You may get cut. Either way, you're starting your day to do all the things you needed to do that are going to put you in harm's way with infectious diseases. So that's why your immune system would be upregulated in the morning. And oh, by the way, there's studies that show vaccine response is better in the morning if a child or an adult receives vaccines between 8 and 12. So we now know circadian biology is massive, right? It upregulates genetic function. So, for example, it turns on genes that are related to how you could process a drug, how you could process a food, how you could process all kinds of different things. So the reason I'm belaboring this point a little bit is because I think we've gotten away from understanding what circadian needs are. It is super important that we are out in the real world getting sunlight exposure when the sun is coming up in the morning and when the sun is going down in the evening. That's what sets the regulation in our brain, suprachiasmatic nucleus. It comes in through the eye and hits that part of the brain in the back and says, hey, okay, set, up, down, up, down, up, down. that piece, it's this huge interwoven web of understanding that that's the biology that's going to help us function at our optimal self, right? So this is huge understanding. And I really want that to be a part of the take home from this. When I think of the pregnancy state and I think of potentially stressors in life and what could be going on, the circadian biology again comes into huge play. Recent studies have shown that if you are mood prone to disorder, light in the morning is critical to reducing your risk of mood dysthymia. And again, this is all tied to immune function, right? Mood is tied to immune dysregulation, or sun exposure is tied to immune dysregulation. So they wouldn't be surprising if they're both tied to each other. So ultimately, we would want to make sure we're getting adequate sun exposure in order to, in the morning, set the rhythm circadianly of what the system's supposed to do from a neurobiological endocrine immune perspective. And then I want to look at some work by Dr. Robert Dahlman, Dallmann. He's a chronopharmacologist, right? So chronopharmacologist, right? So chrono-circadian, following the clock, chronos, pharma, drugs, cologist. So he studies this stuff. So he's involved in understanding how drugs are related to the time clock, right, of the sun and the moon. So he basically studies the effects of drugs on mammals based on the clock of 24 hours of our lives. His pioneering research was recently published in 2014 in the Journal of Biological Rhythms and looked at a mouse model exposed of poisonous doses of acetaminophen or Tylenol and looked at it based on the circadian timing. His research group found that when you give large doses of acetaminophen in the morning, the pharmacological dose was identical, but because of the timing of the day, the enzymes were upregulated in the morning, the death occurrence was mitigated. Right? So this is huge. Think about this from a pregnancy perspective. If you're a pregnant mother, and you have a headache, or you have a febrile newborn, and you give them acetaminophen in the evening, you have a much different response epigenetically than you would if you gave it to them in the morning. And so it's always a question to me, how careful we need to be about using these pharmacological agents, or any agent for that matter, based on the sun and the moon. Because it's very clear that the morning is when things were expected to happen biologically from a systems biology perspective over hundreds of years of humans living on this planet. I think that's sort of the key to this. So for me, it's a big one. And, the take-home point for me is avoid events that would jack up your circadian rhythm, right? Don't stay up late. When the sun goes down, dim your lights, maybe use red lights. Do what you can, dim your screens, read on books, anything you can. Definitely get your kids off the screens when they're older. But again, this talk is about the growing mind. So we're talking about primarily about the pregnancy state. So start working on unwinding your natural inclination to stay up late if you do and sleep in the morning and not get that dawn and dusk understanding of light exposure. I think that's sort of critical. I wouldn't wear sunglasses in the morning and in the evening. I think sunglasses at the height of day makes sense, because you're blocking those really intense light rays, but you can look at the sun in the morning and you can look at the sun at the sunset. And that's when these differential light rays are coming in that are really important for turning on melanopsin and the different functional parts of the brain that come in through the eye that gives us our circadian biology. I think that's super important. To me, drugs are a no-no in pregnancy unless you have no choice, right? Do everything you can to avoid using drugs. And then ultimately, I think biologically we're expected to eat in the morning and in the afternoon and maybe eat less at night, especially after the sun goes down. I would try and avoid that reality in ourselves, really try not to eat late at night. And then metabolic clearance of waste in the brain. I think this is another piece I'd push on. Sleep deprivation is not good for that. So if you're not sleeping enough, you're not clearing the brain through the glymphatics, right? The glymphatics are the lymphatic systems of the brain, where it's called the glymph. And when you sleep at night, especially during deep sleep, when you get into those deep phases of sleep, or even potentially in REM sleep, mostly in deep, you actually clean out the debris of the brain, whether it's mitochondrial debris with oxidant radicals or chemicals that are bound to glutathione or even bound to different lipoproteins. They can get cleared during that period of time. So sleep deprivation would disrupt that. So not only you increased inflammation during circadian biology, but you'd actually increase inflammation due to the inability to clear. So I think clearance is super important here. So thinking about the sleep from the clearance of biological perturbations. And then ultimately, I think we need to start to ask ourselves the question, is there a way to get ahead of all of this stuff? In this conversation, there was a lot of information, and we could go deeper into any one of these topics. But in order to get a global 30,000-foot view, you can't do that. We can get granular at some spots. We got down to 100 feet at some of these, but the vast majority of this is from the big global perspective of multiple holes in the dike. What can we do? Well, for me, predictive value of early autism detection model by that Duke study, again, I keep coming back to that, says, okay, we need to get upstream. Upstream is pregnancy. Upstream is frankly a year before you're pregnant. right? That's where I would like to hit. And I think about the work of Leslie Stone and Emily Ridbaum and their Grow Baby project. And I think that's huge, right? Do SNP testing on mothers, right? Single nucleotide polymorphisms. Then based on that, understanding, make targeted decisions for each person based on macro and micronutrient needs, stress reduction, exercise, all of those levers, as many as you can pull on as once, and then see the outcomes. And their data is pretty cool, right? They're not seeing autism rates like the rest of the country. So for me, that's the work that I'd start to say we should pull on. Really start to get in a global picture, 30,000 foot view, understand at the microscopic level where you can, but in the absence of that, make decisions based on what makes the most sense, right? So if you ask a chemical company, is there a chemical harming you, BPA? They'll say no. And you can't say with 100% certainty that it's not, but I can give you some pretty good data that says that it's not good. The question is it the main cause? No idea, but it's not good. So avoid it, avoid it, avoid it, avoid it, avoid it, and say that avoid it 1,000 more times. Ultimately, what do we want to do? The to-do is the big piece in all things, right? So in the pre-pregnancy phase, you want to focus on immune health with consistent sleep, stress reduction, whole foods diet, organic, as much as you can. Use EWGs, Clean 15, and Dirty Dozen to understand which foods have more pesticides if money is a concern. Toxins need to be avoided. I would encourage 30 minutes of direct sunlight daily based on your skin color without sunscreen, right? Get your sun exposure for your natural vitamin D. That's your tolerance induction through FOXP3 or 4-Kid Box 3. That's how you get your Tregs. Again, tolerance is huge. I would highly encourage a prenatal vitamin every day. I like Garden of Life's. That's what I tell all of our teenage girls to start. Garden of Life's prenatal vitamin, okay? Treat all maternal diseases aggressively before choosing to have a child, especially if you have autoimmune issues. Really work on diet, all the things we're talking about to get autoimmunity under control. Get your diabetes under control. Get anything that's driving dysregulated immunometabolism under control. Treat all the things that you can during pregnancy now, not just the pre-pregnancy phase, but during pregnancy. Go through and do some of that stuff as well. Deliver vaginally when possible. Consider fecal microbial transplant through fecal swabbing. Postnatally, breastfeed, 100% breastfeed, breastfeed, breastfeed. Just breastfeed if you can. It's so important. Definitely encourage everyone to work super hard on keeping the child exposed to high quality breast milk with all the human milk oligosaccharides. Consider a good quality probiotic like Avivo, the B Infantis, or some other variation thereon. As soon as the child's born, I highly encourage, again, breastfeed exclusively till 4 months, but then begin the process of giving fruits and vegetables. Vegetables first, then fruits, then beans, nuts, seeds, all in a non-chokeable form. I'd add the meats and fish and everything up until a year of age. Make sure all the proteins are given in that first time. Look for food intolerances, especially milk protein intolerance in these little kids, right? It's a huge issue in our clinic. Casein intolerance is a major driver of inflammation, and that's going to lay on so many factors that we talked about, whether it's micronutrient absorption, inflammation in the gut, dysbiosis, all across the board. In the kids, again, whole foods, organic as best you can, minimizing starches, promoting phytonutrients, avoiding chemicals as best you can, targeted micronutrients in kids as best you can. If you can get lab work, even better. Spend lots of time outside in nature, teaching your kids how to live and love this world as best they can and learn. And then be attached to your kid as best you can. Be present moment with them, love them, take care of them, give them hugs all the time. It's food for the soul for you, and it's food for the soul for them. Ultimately, it's a web, convoluted, beautiful, but it needs to be looked at. And we need to look at it from the beginnings of time as it relates to your desire to have a child. If you are a mom or a father involved in a situation like this, work hard well before the child is expected to be conceived, and then work super hard during pregnancy and hard afterwards. It's a beautiful thing. So that's my look at the growing brain. Again, I'm Dr. Magryta. I appreciate your time, your love and passion for kids and yourself, and look in the mirror and love yourself so much that you're going to do the work to be the best version of yourself in this world. Be as immune solvent as you can, and just take care. Have a wonderful day.
Welcome to Rupa Health, the best place to order, manage, and track results from over 30 different lab companies in one single place for free. It's going to take you under 2 minutes to sign up, and you can order any functional medicine lab for your client in under 30 seconds. Let me show you how it's done. So here's our beautiful interface. I'm typing in the name of my client, selecting the lab that I want to order for them, and hitting send. From there, Rupa and their amazing team handle the rest. They email the client, collect payment, and even offer an interest-free 3-month payment plan. We've also built the world's largest library of information about chronic health conditions, the lab tests that can help you find the root cause, and the evidence-based interventions that you can use to help people heal from them. It's called the Rupa Health Magazine. There, we have in-depth articles about almost any health condition you can imagine. And we give you step-by-step protocols that other clinicians have used to help their clients heal and that are verified by evidence-based sources. You should totally check it out, and it'll transform your practice. And we can't wait to see you. So make sure you sign up today at rootthehealth.com.